And sitting here through 7 lectures already. We did a lot of mathematics, and today we can benefit from everything we developed and look at some pretty pictures and some applications. So here's a procedure again, just to remind you. We are interested in the macroscopic equation, and then this UC, the drift velocity, and the diffusion tensor, they depend on a function which I call Q, and this was a distribution of directions, basically. And then the drift is the first moment, and the variance-covariance matrix gives the diffusion term. So I talked about the von Mises distribution last time. And so now let's look at the first application, which is Wolf movement. And I indicated this problem already. And so here you see a picture, which actually I took from an aeroplane of a forest landscape in northern Alberta, in Canada.
You took that?
I took that, yeah. I took that myself. So there was a late spring, so there's still snow. So here you see some roads, okay, but then also you see very fine lines going through, and these are the seismic lines which I mentioned. And actually there are many more than you think. So here's a whole bunch, and here, and so we digitized this image, and so these are all the lines in the previous picture. So there, if you compare, there's actually quite a lot. And so these lines are typically 5 meters wide, and then this domain here is about 2 kilometers either way. So what we then do, we take this Q, the movement probability for the wolf, to be these lines. So there's a higher probability for a wolf to stay on the line, so that's the von Mises distribution. But there's also probability to go off and go into the forest, so this probability is non-zero. And it has actually been shown by Mackenzie and Lewis that the von Mises distribution is a good estimator for the movement behavior of the wolf. So this from GPS data. So here's a von Mises distribution again, same picture I've shown last time. And then we compute the moments. So the drift is 0, the diffusion term is this. And then we solve the fully anisotropic diffusion equation. Often in the literature, people assume that the diffusion tensor is isotropic, has an isotropic part and a directed part. And now, based on our theory, we can identify kappa 1 to be this and kappa d to be that. So people would have just chosen random values for kappa 1, kappa d. But now, based on the biology, you can actually find explicit values for these terms. And then here's a simulation which is done by my collaborator Kevin Painter. So initially we have wolf randomly distributed in this domain, and then we let them just move around. And after a short time, T10 is pretty short in this simulation, we see already a strong aggregation along these seismic lines. Between the lines we are in the green area, which is about here. So this is 1, and on the lines it is 2. So the density is not 0 in the forest. You also find the wolf in the forests, but you find them more often on these lines here. Okay. Then we looked at the home range of the wolf. So wolf packs usually have a home range where they hunt, and we assume there's a den site somewhere in the middle. And then we look how the wolf would spread if the lines are present or not. And so here you see how they would spread if there would just be forest. But then with the lines, they actually extend much further than they would do otherwise. And this can then explain why they hunt better, or why they find more deer, because they're basically extending their hunting ranges quite significantly. Okay. So the second that's what we have done for the wolf. So next would be to put the prey in there, so the deer. And then the deer might also use the lines to get away quicker, or they might avoid the lines because they are the wolf. So if they are smart, they would maybe try to hide out somewhere in the middle. So different behavioral strategies can then be tested with this kind of model. Okay, so glioma. So a quiz to you guys. So where is this picture? Who knows? E2, exactly, right? So it's right here on campus. If you are from KAIST, you have seen it probably every day. If you go up E2 to the 3rd floor. So this is what I use as well, these kind of brain images. So Yeah, let's talk about glioma a little bit. So glioma is the most severe brain tumors which they are. They're called glioblastoma multiforme. And often they are found of this size, maybe inside the brain. And they are found if something serious happens. So people get blind, or they have a lame leg, or they can't breathe anymore, or whatever. Something goes wrong in the body. And this is the first indication of a brain tumor in some cases, not always. But then if it's found on this size, it's already very, very serious. And then these patients are treated. So this region here is maybe taken out, removed surgically, and then there's also some radiation applied to the surrounding tissue. And then the survival is about 12 months to 14 months, 12 to 14 months. So there's only 1 year survival expectation. But also, these patients get severe brain damage, right? So anything can happen. They can be in coma, they can be paralyzed, they can become insane or whatever. So it's really terrible. And sometimes you might even wonder, is it worthwhile to take it out, or maybe just let the person have some decent life, and then the life ends. Okay. So one issue though is. What you see here is not everything. So it is known that the tumor has spread more than what you actually can see in the images or in this image. And it's very important to try to get the tumor cells which are hiding away from the central tumor mass. And this is what we like to do with our modeling. So we would like to predict how does the tumor spread further than what is visible. Okay. And what we use is the anisotropy of the brain. So I talked about this as well. Here's another picture of the brain fibers, or the white matter tracts. And actually this dominant fiber going from the front to the back, actually I don't know where the front is. I think here's the front. So this dominant line through here, this is the visual nerve fibers. So it goes from the eye to the visual cortex. So it's one of the biggest fibers in the brain. But then there are also some which connect the hemispheres left and right, here and here. And then there are a few smaller ones, say, on the outskirts here. All right. So the nerve fiber tracts, they look like this. So this inside would be a nerve fiber, and this is surrounded by some myelin, which is like an insulation, like you have insulation on a copper wire. Yeah, and then they bundle up, and they can form these nerve bundles. And then tumor cells, they would kind of move along these nerve bundles and invade. And a cross-section is then this, where you have the insulation, and then inside is the nerve fiber.
What's the thickness of the fiber? One fiber?
There must be in the range of mu meters.
Millimeters.
Mu, so 10 to the minus 3 millimeters.
It's
Yeah.
something like the comparable size to the
Of a cell.
cell.
Yes, that's right. Yeah. Actually, this insulation thing you see, these are cells. These are cells which are wrapping around the nerve fibers. Yeah. And well, since I'm talking about this, so for example, you might have heard about multiple sclerosis. That some people are infected on this, and in multiple sclerosis, these insulations are attacked by the immune system, and they're eaten away, right? And then if you have fiber nerves together where the insulation is gone, then you might have short circuit, and things don't work anymore. So just on a side remark. Here are some closer ups. So there are some connections between these myelin sheets along the nerve fibers. But then what you can do with tractography, you can try to visualize these nerve fibers in the brain. So this is one of these typical images. So if you type tractography into Google, you get millions of these kind of images, where people try to reconstruct the nerve fibers in the brain. So that's a brain domain. And not all, but many of the nerve fibers are kind of shown, and to try to understand how the brain is connected in different areas. Okay. Then there are also individual studies where people really follow individual glioma cells along these fiber tracts, and you can see how they align with the underlying fiber tract. So one way to measure this, and this is important for our modeling, is diffusion tensor imaging. I mentioned that also, just roughly. So here's the brain domain, which is divided into a grid, and at each grid point you have an ellipsoid, which then is of course a diffusion ellipsoid, which I have talked about already. So you have a dominant direction, maybe lambda 1, and then secondary directions V2, V3. And this can be very cigar-shaped, very anisotropic, or it can be very round, which is then isotropic. And so now we like to use these data to infer the connectedness of the white matter tracks and then to put this into the tumor growth model. So here's the main idea. So we have the brain domain. We get the diffusion tensor imaging, which are these ellipsoids. This is the measurements. We make some conclusion about the alignment of the white matter tracks at those locations. And then we put our cells into this fiber network and let them walk. So these are not fried eggs, so these are cells. So the cells move in this network, so this cell would move more diagonally, for example. And this cell here could move in any direction. And this gives us the Q. So now we have this variable Q. We can do our moments. Second moment is a diffusion coefficient and solve the macroscopic model on the brain domain. And then we get a prediction for the brain tumor. So there's a lot which goes into this, in particular here for the movement on the fiber network. This is what I talked about in the previous lectures. So we obtain the measured DTI data. We define Q as a von Mises distribution based on these diffusion ellipsoids. And then we compute the diffusion tensor dt as a variance-covariance matrix of Q. And then we are in business, and then we solve this equation. And I put in logistic growth because the tumor is also growing while it expands. So we also have a growth term here. But the key issue really is the anisotropic diffusion, which shows up there. So let's see how it looks like. This is a simulation done by my PhD student Amanda Swan. She will finish this summer. And the data are from the Cross Cancer Institute, which is a cancer clinic in Edmonton, and Dr. Al Murta, so he has all these DTI data. So this is one slice of a brain, and underlying, which we do not show, is this DTI information. So at each point you have an ellipsoid, which tells you in which direction it would grow. And this is a healthy patient. I think this is even the data from Dr. Murta himself. So, but then we implanted him with a tumor. and now I show you how this tumor would grow in that irregular brain domain. Yeah, so you see it does not grow like a round-shaped kind of thing, so it grows very irregular, and you get lots of structure, right? You get higher densities here, and then lower densities here, and then higher densities here. But then also on the outskirts, you see that maybe it has found a white matter track and going down here, where it's maybe not so much in this direction. So if now a clinician comes and just takes a circle or a uniform circle around here, then they would treat regions of high tumor, but they would also treat regions of low tumor, and they might even miss regions of high tumor density. So this is kind of the information we are after. That's what we like to see. So we started with the validation. So now we are taking existing brain tumor patients which do have a tumor, and in this case the real tumor is indicated in a black outline. So the black outline is a tumor as was indicated by hand on a computer screen by a clinician. And then the white outline is our prediction from the mathematical model. And you see that this overlaps very, very nicely. So it's certainly accurate up to the grid size, which we have. But there's more information in it. So now the color scheme, red, yellow, blue, and light blue, dark blue, this is now the kind of the concentration of the tumor cells, or the level set of the tumor cells, as you will. And the white outline is the 0.16 level set, which corresponds to the resolution of the MRI scans. So this would be the region which would show up of an MRI scan, but the yellow, the blue, would not show up on any scan because the density is too low. But now from our model, we can maybe say it has grown more here. It has not grown so much in this direction. It has grown more here, and so on. So this is kind of the information we are after. Okay. So then we can use this for treatment planning. So then the idea is to give clinicians an idea, maybe where should they treat more and where can they treat less. And here are two patients, patient 8, patient 3. Let's look at the left one first, patient 8. So black outline is the actual tumor, and then white would be the clinical treatment region. This would be the region the clinician chooses to treat. The red line is our computed treatment region. And so here in this case you see they're basically identical. So everything is good. On patient 3 it's different. So the clinical treatment would be the white outline. Our predicted treatment volume would be the red line, which is very different, right? So we would, in this case, we would suggest to treat less here and to treat more here. And the reason is that there are white matter tracks which are connecting this area to the tumor area. So this is just the first try. This is, I hope no clinicians uses this really. So we need to do much more validation and be much more sure that what we compute here is really, really sensible. But that's the direction we would like to go. And if a change treatment modality maybe buys a patient another half a year, I think then it's worthwhile to do it. That would be okay. So the real tumor lives in 3 dimensions, so we can do the same thing in 3D. And I've shown you that we have the von Mises distribution moments in 3D now as well, which we need. And so then here is one case where the segmented tumor, which means what the clinician says is a tumor, is this, and our simulation is this. So we are certainly in the same kind of bulk part, but also we seem to have missed some structure from the tumor. And then here's another case where this is the segmented, so the clinical tumor, and this is our predicted tumor. But also what you notice is you have kind of a terrace kind of structure, like rice terraces in China. And the reason is the slicing of the brain from the imaging. Right, so you can really see that these are different brain slices, 1, 2, 3, 4, 5, 6, 7 slices. And then the clinician took every slice and rendered what is the tumor, and then takes the next slice and renders. So it's not a 3-dimensional rendering. And this could also explain why it looks so staircasey here, and in our case it doesn't. Yeah.
So it could be this way then.
So that could actually be more accurate than this one. I mean, here's a plausible explanation why it has this structure, just from the processing of the data. Yeah. So this is certainly not the end of the story, it's just the beginning of the story. So there are many details that need to be worked out and need to be understood how this one relates to the other, etc. Okay. So, and then maybe before I do this, maybe do you have some questions about the glioma stuff?
How about the imaging of the fibers? Is it all different people have different images?
Well, there are some dominant nerve fibers which everybody has the same. Like the eye, from the eye to the visual cortex is the same for everybody. Yeah. Or connection from left and right hemispheres is the same. But then the fine structure is different.
So if they are not that different, then you may use just one image for each patient.
Very good idea, yeah. So, no, it's right. So there's something is called an atlas. There's a brain fiber atlas online, which you can download, and this is kind of the typical connection which you have in a brain. Yeah. And this is used. It's particularly also used for these kind of models. But it turns out that each person is different from each person. So if you really want to treat people, you need to look at the individual pictures. Yeah. No, but these atlas exist. There's also symmetry between left and right. So if right is damaged, you can look at the left side of the brain and get an idea about the connectedness. Okay. Other questions for the glioma? Okay. And then the next one is cell movement in fiber networks. This has to do with melanoma. So melanoma is now brain cancer. Skin cancer. And in skin cancer, the fiber network is made out of collagen fibers. So the skin is mostly collagen, and then the cells move in this. And actually, this was the first case we studied. Now it's the third application, but it was the first case because here the data are best. Here we can follow individual cells. So people can take a piece of skin, put it on a petri dish, put in a cell, see how it moves, etc. So I show you a few pictures. Here's a sketch first. So this is one of these melanoma cells, and here are the collagen fibers. And they move along these fibers, but they also cut them. They release a proteolytic enzyme, and then they cut the fibers where they came through, and this forms a tunnel. It forms like a tunnel defect, and then other cells can come through this tunnel, and this acts like a highway. So then other cells can maybe invade the same route as this initial cell did. So here I show you one of these moving cells. Do you want to work today or not? Let's see. Options, trust always. So. And there we go. So it's a short movie. I might want to show it twice. So here you see one cell. This green thing is one cell, and this is part of the same cell. It just goes out of the plane of focus and comes back in. And the gray background is the collagen fibers. And then here you see a little bit that there was cutting going on. There's some kind of fibers missing if you look very, closely. And yeah, let's show this again. But you see, so it's pulling on the network and then moving through the network. So it's modifying the network, it's cutting the network where it came from. It's also a very elongated cell. Yeah. And this is called mesenchymal motion, so when they get very elongated. Now let me show you what this looks like in a real tumor. So now this video is even shorter. The screen will split in two parts, and then you see a tumor sitting here on the left and another one here sitting on the left. And then you see individual cells kind of shooting out of the tumor and then releasing a trail where they have cut the fiber network. And you can sometimes see other cells following behind. So I show this also twice. This was short, wasn't it? Shorter than any commercial. So, okay, so here you can make out an individual cell, very elongated. Here's the trail where it came from, and here's already another cell following it and using the already established trail to invade. In this case, the cutting abilities for the cells have been inhibited, and so the spread on the same time scale is much less because they can no longer cut, but they can still make their way through. Okay. So we like to have a mathematical model for this, and of course I use a transport equation. Again, because I have the information about the collagen fibers, and I would like to track the cells. So theta is the fiber orientation. Now fibers can be undirected or directed. So collagen, which I'm interested here, is undirected. So at each location along the fiber, I cannot tell what is up or down or plus or minus. There are other fibers in cells like microtubules or actin filaments which would be directed. So in that case, at each location, you know what is plus or minus, or active, passive, or however you call these. So, so we are in this case, and then we have our distribution of fibers Q. So we are in business, right? Yeah, Fv is a set of cell velocities. So we discussed all this in great detail, and then I take the distribution of fibers and I lift it to become a distribution on V by a scaling parameter omega. So we have done that as well. And so then I come to the transport equation for the cell density, where the fiber distribution Q shows up here. And so that's the same transport equation I've talked about before. So it's really cool. So now I know that you all understand what's going on in this equation. Okay. But now Q, the fiber network, depends on time because we have the cutting. So the cells interact with the network. So what I need now, I'm not using a von Mises distribution now. I need a differential equation for the fiber network. So Qt, time derivative, is something which depends on the fibers and on the cells and describes the cutting which is going on. So we do it like this. So maybe I'll show you the next slide. Yeah, that's better. So this is the distribution of fiber directions. And so this operator, this pi minus a, should describe the cutting. And you cut if a cell comes together with a fiber, so this is kind of a mass action kind of term. So the real dynamics happened with this pi minus a, and it goes like follows. So here I plot 2 cells and 3 fibers, and the cells have velocity v and the fibers have direction sigma. And I look at the projections of the cell movement onto the fiber direction. So here I plot all these projections, okay, on all the fibers, and this is the operator pi. And then I take the mean length of all these black lines. So the mean length is a length which I call a. And then I compare the projection with the mean length. And so if the projection is longer, there's no cutting. If the projection is shorter, then there is cutting. And then with a certain probability. So there's no cutting with a high probability, and there's cutting with a high probability. So that's in a nutshell how the model works. Okay, so then I get a system. I get the transport equation here, and I get the fiber network dynamic equation here. And now that's a very interesting system, and we would like to see if it does what it should do. So having cells move through fiber network, cut, etc., etc. So I show you a simulation done by Kevin Painter. We're in Initially we start randomly with random cells, random orientations, random orientation in the network. And then you see while the cells move about, they form roads and highways just by this cutting mechanism, and other cells aggregate there, and then they tend to follow these network structures as well. And so then the time is still running, but numerically there's no more change. So they look like steady states. And this pattern formation is very robust, so this can happen on many different scales. But it's kind of cool. So it really explains nicely how this invasion happens out of the tumor, and then other cells can follow and can form these highways, etc. All right. Okay. So I talked a lot about the parabolic limit. So here now we have kind of a new situation. We have a transport equation coupled to the fiber network. So what about the parabolic limit? Can we do that? Does it make sense? And we can do it, but we lose. We lose a lot. So that's why I like to show here in simulations rather than in formulas. So what I've shown you is maybe somewhere here or here where you get these network structures. And then I look at 2 cases. One is diffusion dominated, which would be the parabolic limit. The other is drift dominated, which is the hyperbolic limit. Right? We had both cases. So in the parabolic limit, it turns out that these structures, if you go further to the limit, get finer and finer, and in the limit you lose them. Okay, so in this case, the parabolic limit equation can be computed, but it becomes a diffusion process, and you lose all the structure, lose all the information you are interested in. In the drift-dominated case, you get, so here's a network alignment. You kind of focus in on one of these roads, and then you just get a completely aligned tissue out of this. So you also lose the information. So for this problem of the skin cancer cells, the intermediate region is the region we would like to be in. So the diffusion limit exists, but it doesn't tell you anything. The hyperbolic limit exists, but it doesn't tell you anything either. Okay. All right. So I think I'm maybe a little bit early, but I think nobody will complain after 7.5 lectures. So let me just conclude. A few conclusions here. So for the transport equations, we have seen 2 cases when the eigenfunction is constant and when the eigenfunction depends on v, and they used a little bit different methods. Now on the first case I spent 8 lectures, and on the other case I spent 2 lectures, but I think you understand that it makes sense. So I wanted to have one case which I can completely describe in all glory details, and I think it makes a lot of sense to do that, but then I didn't have time to do this case in so much detail, but that's probably okay. And you got my notes, so both cases are in these notes. You can look it up. We looked at scaling limits, parabolic limit in much detail, hyperbolic limit, moment closure in less detail. Again, for the same reason, I think it's important to see one of these methods really, really well. The macroscopic model can be useful. For example, for wolf and glioma, I think it's really useful in that case. Or insufficient, for example, cells and fiber networks, it would probably not do a good job. Okay. So what are open problems? Mathematics, convergence results. So I used a lot of scaling arguments, relating models here and there, forward, backward. Most of these scaling arguments I've shown are not proven rigorously. So if you want to get your fingers dirty, do some interesting mathematics, you can look at these limits. So many of them have not been proven. Steady states of the kinetic model. So I've shown you the simulation where these networks form. And I think these networks are steady states, but how to characterize them? So these are complicated, spatially inhomogeneous steady states. And so we had a few ideas, but it's widely open what are the steady states here. And then the qualitative analysis, how the pattern formation arises, or even the linear stability for the last model I have shown is all open. This can all be done. Applications. Yeah, for the wolf, we want to include the prey, predator-prey dynamics. For the glioma, we have many more data, so I now have a postdoc, and she works on getting the model to the patients. So this is one driving motivation for me now. I want to carry it as far as I can for patient treatment planning. And then another application, sea turtles, I will talk about next week at NIMS. So actually, when is my talk?
Next week, Thursday.
Thursday probably, yeah.
4:30 or 4:30.
Okay, something like this, yeah. So I need to leave a little bit for just another talk, so that I'm not including it here. All right, so right around Christmas last year, I was walking on campus and everything was white, and there was something shiny in the snow. So I bent down, I picked it up, and it was an old lamp. It was very dirty, so I rubbed it to clean it up. And so smoke arose, and then Kim Young-jung came out. And he asked me, why don't you come to Korea and give a series of lectures? And so I'm here. Thank you very much.